I myself, Shruti Tiwari, a student of MSc second year program, is going to present a small talk on how mutation could cause behavioral changes. Here, I will be talking about the current state of Drosophila behavioral genetics by focusing on a set of specific examples and by deriving lessons that might be of general significance to the question of how genes affect complex behaviors such as courtship behavior in Drosophila. The talk centers on a discussion of how Fru gene contributes in the courtship behavior of Drosophila and what behavioral changes are observed due to mutation in that gene. What is mutation? Mutation refers to any sudden heritable change in the genotype of a cell or an organism, providing them with new variations that leads to the evolution of the organism. The term mutation refers to both the change in the genetic material and the process by which the change occurs. An organism that exhibits a novel phenotype resulting from a mutation is called a mutant. What are behavioral mutations? A category of mutations classified on the basis of its phenotypic effects, or say that affects the behavior patterns of an organism, is termed as behavioral mutation. For example, the mating behavior of a fruit fly. When genetics meets behavior, the challenges of behavioral genetics research include the difficulty in defining and quantifying the behavior, the tremendous within and between individual variations in behavior, the involvement of many genes, and the fact that different genes function in different tissues at different times during the ontogeny of an organism, all of which combine to influence a single pattern of behavior. In addition to these genetic contributions, an organism also experiences environmental conditions throughout its life that can influence its behavior. Here, I am using the Drosophila as my genetic model organism. Model genetic organisms such as Drosophila have been especially useful for the genetic dissections of developmental and anatomical traits. Why? Because the fly have a structural or functional homology in vertebrates, including humans, and it can contribute to our general understanding of evolutionary, conserved developmental, and physiological processes. And it also is a very very exceptionally very good model for the study of simple and complex behaviors such as courtship behavior. Uh, in this slide, I am showing the Drosophila melanogestures courtship behavior between males and females. Drosophila male courtship behavior is species specific. At the beginning of courtship, the male stands facing the female, called the courtship orientation. The wing display usually follows after a few seconds of orientation. One wing is brought out until it is at right angles, uh, as you can see here, to the male's body. Then it is vibrated rapidly up and down for a few seconds. And thereafter, the male comes behind the female and licks her genitalia with his proboscis and attempts to copulate. It appears that licking is always followed by attempted copulation unless the female is able to prevent it. During vibration, the male is always orienting, and during licking, he is both orienting and vibrating. These three elements, the licking, orientation, and vibration, are not distinct but are superimposed upon one another. This figure gives a representation of this patterning. Talking about the fruitless gene and its involvement in courtship, the fruitless gene, Fru gene, was originally identified on the basis of the aberrant courtship behavior shown by Fru mutant Drosophila melanogester males. It is a member of the Drosophila sex determination cascade and belongs to the BTB zinc finger family of transcription factor. Okay. Uh, Here are some more genes in addition to the Fru gene listed that play important role in courtship behavior, and their behavioral pleiotropy is also shown here in this table. This slide is of genomic and transcript map of Fru locus. It has the Fru gene has four promoters P1, P2, P3, and P4, but only the pre-mRNA from the P1 Fru promoter is spliced in a sex-specific manner, and P2, P3, and P4 have no sex-specific functions. The Fru gene also has pleiotropic effects, as I said earlier, and is responsible for a range of phenotypes, some of which affect male courtship behavior. A particular set of Fru proteins that are generated from transcripts associated with the P1 promoter are responsible for the role of Fru in courtships. And Fru seems to be required for almost all steps in the courtship behavior. Okay. Fru gene expression and signaling. The male specific Fru gene transcripts are expressed in the central nervous system. The cells that express Fru transcripts are not localized within one substructure because approximately 20 groups of cells that express the male specific Fru gene transcripts are distributed throughout the CNS in both brain and ventral nerve cord. The study suggests that male sexual behavior arises as sensory information is transmitted to the CNS through non-sex specific sensory systems. This information is then processed by the higher order neuropil. Neuropil are the cells that express Fru gene in a sex specific manner using the putative sex specific circuitry in which Fru acts. Subsequently, sexual behavior is possibly initiated through non-sex specific motor systems. Identification of the circuit that Fru expressing neurons might form is one of the next challenge for Fru researchers. Next, talking about the mutation in Fru gene, both viable and lethal Fru mutations exist here. The viable alleles alter male specific courtship functions. Severe viable alleles of Fru generated by transposable element insertions almost completely abolish courtship behavior and cause aberrant splicing of the sex-specific transcripts. A weaker insertion allele, Fru2, produces a different aberrant splicing pattern and results in problems with song production. Here, as you can see here, the Fru2 gene. Okay. Another viable Fru mutant stops the courtship sequence during sperm transfer, and another Fru1 results from an inversion that alters the spatial pattern of gene expression in the brain. When Fru mutant males are grouped together, they form male-male courtship chains. That is, each male is simultaneously both courting and being courted. As you can see here in this diagram, fruitless mutants are also observed to have a muscle of Lorentz defects. There are other mutations too in other genes other than Fru in Drosophila, such as dunes, rutabaga, and amnesiac, that affect CAMP signaling and that disrupt olfactory-based avoidance, associative learning, also alter courtship suppression in Drosophila. Modest reduction in calcium or calmodulin dependent protein kinase 2 and protein kinase C, two kinases known to involve in neuronal plasticity. They also alter different aspects of courtship suppression. So, different signal transduction pathways have overlapping roles in the experience dependent modification of courtship behavior. Conclusion. Although I have discussed only one aspect of how genetic mutation affects behavior, there are many unknowns in the puzzle. The powerful array of genetic techniques in Drosophila and its well established place as a model organism in developmental genetics provides researchers with a unique opportunity. That opportunity is to create a comprehensive picture of how continuing interactions between the genome and environment select for naturally occurring mutations that can act on specific neuronal networks to modify the development and or functioning of behavior. No doubt, Drosophila, with its array of genetic resources and behaviorally based screens to identify new genes involved in conserved pathways, is at the forefront of behavioral genetics analysis. Undeniably, this little fly will continue to provide us with many tantalizing discoveries. And these are my references. Thank you.